So everything that's happened to you since you've come into this room is a result of information that has come in through your sensory systems. The people that you've recognized, the conversations you've had, the bitter taste of the tonic in your gin and tonic, the smell of the wine, have all come in through sensory systems, and in fact have for your entire life. So everything you know about the outside world is really a consequence of your sensory systems. And these sensory systems have a remarkable challenge. First, they have to work at the very threshold of detection. The visual system can detect a single photon of light. The auditory system can detect sound at incredibly low concentrations. The olfactory system of a mammal can detect odorants at concentrations as low as a few parts per trillion in air. And mosquitoes, one of the people here, Chris Potter, wherever he is, works on fruit flies and mosquitoes, and we both all well know how their amazing ability to find us and our food at the most inopportune times. And so they work at the incredible threshold of detection. At the same time, they sit right out at the sort of frontier of between ourselves and the outside world. And as such, they are constantly being assaulted and insulted by either intense stimulus or alternatively by pathogenic or other environmental insults. And so we've been very interested to try and understand how these systems have taken particular adaptations in their ability to achieve these balanced, remarkable capabilities. So in the Center for Sensory Biology, we want to know what's the mechanisms by which sensitivity and specificity or selectivity is achieved. Secondly, how do these systems adapt and protect themselves from an incredible spectrum and breadth of stimuli, from the softest sound to the loudest rock band. And then at the same time, how disease in a variety of ways impacts to reduce sensory function and in some cases eliminate it. Loss of hearing, loss of olfactory acuity, loss of sense of taste, et cetera. So those are really the functions that we really have focused on across the center. And when we first started the center, now nearly a decade ago, the thought was that investigators working on different sensory systems, which typically had relatively little interactions with each other, could in fact benefit from a more collaborative environment. And secondly, we could teach each other about particular things, inform and assist, and you may hear an example of that from Paul Fuchs in a few moments. And at the same time, that our work on sensory systems would spill out much more broadly to other areas of medically important biology. And so we began those, and I thought now I'd turn and tell you a little bit about my own sort of experience. I had been interested in the olfactory system from the time I was a graduate student. And Hopkins provided me with a really unparalleled opportunity for the freedom to do research in whatever area I wanted to do. And so I decided I was going to tackle olfaction. And there were really a couple of challenges. The first challenge was that I never worked on it in my entire graduate career or as a postdoctoral fellow. I worked on bacterial antibiotic resistance as a graduate student and worked on immunology, as did Steve as a postdoc. So I came to Hopkins as a new faculty member wanting to tackle a new problem. The second problem was no one knew anything about olfaction at a kind of molecular level. And then finally, the third problem was that it had been described actually about 100 years before by a famous physician as being clearly the sense that was least useful to man in the state of civilization. So this isn't going to get me very much money from NIH, nonetheless, at least these days it wouldn't. Nonetheless, I thought it was an interesting problem, and we then set out to tackle it at a very exciting time in molecular biology and molecular neurobiology. And in the course of a few years, we took the two examples that we knew about how signals were detected in other systems. That was in vision, where we knew, Jeremy Nathans, another one of our center members, had discovered rhodopsin, the thing that detects light and is in your eye so it can detect light. And work by Lubert Stryer, the classic biochemist um, book that many of us used, who had shown that there was a G protein involved. And we decided that that system and how vision worked, and we knew about one other system, that was the beta-adrenergic system. We knew there was a G protein there. And we thought, it's a great system, maybe olfaction works that way, we'll take a hunch. And so we went hunting. And in the course of a few years, our lab and many others as well began to start looking, and we discovered a G protein that was clearly involved in olfaction. I won't tell you how we did that, but it was clearly the one involved in olfaction. But in the course of those studies, we basically discovered a menagerie of additional proteins that were expressed in brain and liver and everywhere else, suggesting that in fact G protein signaling in general was incredibly important. We now know it is in fact the most probably important signaling transduction pathway in humans. And then in parallel, we went on to look for other components in that system, adenylyl cyclase, discovered that those proteins were involved in learning and memory in the brain, and there was a specific form in the olfactory system. And likewise with a couple of other components, and recognized that one of those was shared in vision. So out of this study of this least useful to man in a state of civilization, there were many spillovers that went broadly through science. And in fact, our host tonight was interested in G proteins and G protein signaling in slime molds. And we actually collaborated with her mentor to find some of those as well. So clearly there were lots of things along the way. More recently, now that we've actually had a pretty good understanding of what the common transduction component is, we've been interested, how do you detect so many odors, 100,000 odors in mammals and similar numbers, maybe not quite as high in fruit flies and other organisms. And what we understand is that there in fact is an incredibly large family of similar related proteins called G protein coupled receptors selectively expressed in our olfactory system at the back of our nose. And all of those different receptors converge onto this common pathway. How do we generate specificity? We put different receptors in each cell. And it turns out that in mammals, we have about 1 million sensory cells, sensory neurons, and they fire action potentials, and about 1,000 different receptors, each of which is individually encoded into the genome. And remarkably, each olfactory neuron picks only one receptor out of those 1,000 scattered across your entire genome and activates it for expression in a particular cell. So you have 1,000 different kinds of cells and about 1,000 of each specificity. How the cell does that, we've learned a lot, us and several of our colleagues. In fact, it's a remarkable process. They essentially do a random stochastic selection process, epigenetic process. It's become very popular these days. And then, just to be sure you do it right, do it once and only once, there's a mechanism that shuts off all the other receptors scattered across your whole genome. It is one of the most remarkable epigenetic processes that I think we know about in mammalian cells. So we've learned a lot about that process. Now, in fact, we don't detect odors in the cell bodies of each of these neurons. These neurons sit in the back of your nose. They send out a little process that wave goes out into the mucus and therefore the air that you breathe in. And they send a process all the way back deep into the brain. And that's kind of, if you think about it, kind of a bit of a risky thing to do. You stick something in the outside world and you send the same wire all the way into your brain. It really is like the superhighway from getting from outside to inside. And moreover, every time you smell not wine, but say ammonia, you can do damage to those cells. So we've been really interested in how those sensory cells basically can get assaulted with damage and maintain their connections. And what we've learned is that in fact, they do so by burying their cell bodies down in a tissue more like your skin. But because they have to detect chemicals, they have to send something out. And what they send out are cilia. And cilia, in fact, we didn't recognize for a long time, are like very long spider-like processes. You can see them on some of the pictures as you go through here. And these are the only parts of the cell that are actually out in the outside world. And in fact, all of those transduction components I told you about are actually localized to cilia. And that's where all the initial events, biochemical events, have to happen. Then it turns out that all of the cells that make the same receptor go to the same place in the brain. And as a consequence, when one kind of cell fires, we'll call it the red cell, a little spot in the brain lights up. And your brain basically reads that pattern to know what the odors are. So we were really interested in how all those proteins get out to cilia. And in fact, we used one of those transduction components as our model. And the other thing we've learned is that protein that goes out to cilia to maintain that process is in fact not expressed only in olfactory cells, but in other neurons in your body, and in fact in a few other places like in kidney. And in fact, what makes that particular version of the protein special is that it can go out to these special structures, cilia. And we're now beginning to realize from looking at some genetic diseases in human that in fact putting transduction components into cilia, whether it be olfactory cilia, the most elaborate cilia in your body, or in photoreceptors, also specialized cilia, or in fact in brain neurons, is very important. And we know about a number of signaling pathways, called sonic hedgehog, for example, that must go through cilia to send their signals to the cell and to the rest of the body. And that suggests that in fact, being able to have cilia and being able to transmit signals like neuroendocrine signals for feeding perhaps and behavioral changes might be important for them to come through cilia and provide some new things that we never knew about. We never actually knew that neurons had cilia until we began to look using some of these tools. And in fact, the tool from olfaction turns out to be one of the most popular to find cilia in your brain. So that's a story about cilia, cell biology. And then the last story I'll tell you is one that I think I'll begin by saying if I were in a little black turtleneck, I would say one more thing. And I'm not, so I won't do that. But I will tell you that there is one more thing that makes this whole story even more remarkable in olfaction, at least to me, and was the second thing that really drove me to study this problem more than 2 decades ago. And that was that your olfactory neurons, because they're being constantly assaulted, have the ability to undergo continual replacement throughout adult life about every 60 days. And moreover, if you give them a really traumatic insult, for example, 20 years ago when you weren't wearing seatbelts and you hit your head on the windshield and cut all the nerves that went back to the brain, those conduits I told you about, that all the neurons would die, but within a few days they would all come back again, within 2 to 3 weeks, if you were lucky and didn't have too much scar tissue. Additionally, when you have a cold and you lose your sense of smell, in some cases, what's happening is you're losing your olfactory neurons and then they're regenerated again. All million neurons. And we now appreciate that for cases of sinusitis, inflammatory processes involving a variety of infections, for example, that those cells also can undergo regeneration. But recently we're now exploring the idea, an observation, that in fact those cells regenerate only after the inflammation has been resolved. And we think about teleologically, I hate teleologic arguments, but at least you can imagine that in the case of an inflammation, you would want to not regenerate those conduits back to the brain until you've resolved the inflammation. So we've really focused ourselves on olfaction as this curiosity for the last 2 decades, continuing to do that. But in the process, we've had really just a wonderful trip of being able to see things throw off that have impacted science broadly. And we're beginning to learn something about the principles about why, for example, olfactory systems do this, and Angelica, you hear about in a few minutes, and Paul, that other sensory systems actually take slightly different strategies or lack the ability to do this. And clearly in an aging population, olfactory as well as other sensory modalities begin to fade, as I and others are beginning to notice. And we really need to understand about ways of protecting. And with that, I think I'll stop. Thanks.